Science 2 subject, and we are going to start with the Science 2 period now. So I hope all of you have joined the previous lecture. So we are doing the chapter 16th of Science 2, that is heredity and variation, right? So we'll start with the chapter. I'll share you the screen. Excuse me. Okay, so we are doing the chapter heredity and variation, right? Those who have attended the previous lecture, you know, we have completed the what is heredity, what is inheritance. Then we have also studied about the structure of DNA in details. We have studied about RNA and the different types of RNAs, right? And in the previous lecture, I have also taught you about Mendel's monohybrid cross, right? But students, today in this lecture also I will deal with Mendel's monohybrid cross, because until and unless you understand monohybrid cross, you won't understand Mendel's dihybrid cross, right? Because the dihybrid cross is little bit confusing and difficult to understand, right? So I'll revise Mendel's monohybrid cross, and then we'll continue with the next topic of the chapter, that is Mendel's dihybrid cross. Okay, so pay attention and try to understand. And students, all of you, also all of you can take your notebooks and pencil so that you can solve the cross along with me, so that your concepts are more clear. Yes, so we have seen this pair in the last lecture also. Who is this? This is called the father of genetics, is the scientist Gregor Johann Mendel, right? So Gregor Johann Mendel was an Austrian scientist. He studied the inheritance of characteristics of the pea plant. So he has done all his research, most of the work on a plant that is called pea plant, right? He showed that the inheritance of these characteristics followed certain principles, right? And his work was recognized later. He has done his research in 18th century, but his work was recognized in 20th century. So after reconfirmation of these principles, the same principles now form the basis of modern genetics. So he has developed the laws of inheritance, and now he is called as the father of genetics. He was born on 20th July 1822, students, and he died on 6th January 1884. Okay, so he has done a great work in field of genetics. So I'll show you the plant. This is the plant on which uh, Mendel has done the work. See, this is a pea plant. The scientific name of the pea plant is Pisum sativum. In Hindi, it is also called matar, or in Marathi, it is called vatana. Right, so genetic material is transferred. We know this, that the genetic material is transferred in equal quantity from parents, right? From male and female parents, the genetic material comes in the equal quantity to the progeny. Now, Mendel did not understand, when from both the parents the genetic material are coming in the equal quantity, then whose character is going to be expressed in the next generation. So he has found this out with the help of the visible characteristics in this plant, Pisum sativum. So Mendel carried out research in this direction and put forth the principles of heredity responsible for such inheritance. All of the Mendel's experiments were based upon the visible characteristics of the pea plant. So this is how the pea plant looks. You can see the flowers are there to it. Then we are having a pod of the pea plant. The seeds are there. Then dominant and recessive characters in the pea plant. So what he has said is, he has studied all this and he has said that there are some dominant and recessive characters. The dominant characters are those characters which are expressed in the next generations, right, easily, and more times you can say, or more frequently, these characters are expressed. So he has studied that the shape of the seed, that is the round shape of the seed of the pea plant. The seed is there, right? So if round shape of seed is there, that is dominant character, and if wrinkled seed is there, that is recessive character. That yellow color of the seed is dominant character, green color of the seed is recessive character. Purple color of the flower is a dominant character, while white flower is a recessive character. In this way, the other characters also he has studied. Shape of the pod, that is inflated pod, is a dominant character, and constricted pod is a recessive character. Then yellow color of the pod, dominant, green color is recessive. Then position of the flower, if the flower is axial in position, that is a dominant character, and if it is terminal in position, it is a recessive character. Then stem height, if the plant is tall, that is a dominant character, and the dwarf plant is a recessive character. So these were the seven mutually contrasting characteristics on which Mendel has studied. Right. So this you should know. So as in plants, there are dominant and recessive characters in human beings also. So quickly we'll discuss that. So eye color, if brown and black eye color is there, so it is a dominant character. So you'll find that most of the people, they have brown or black colored eyes, and having blue color eye is a recessive character. That is, it is rarely seen, or very few people have these characters. Then eye size is a large. If the eye size is large, that is a dominant character. Small eye size is a recessive character. In the same way, hair, curly hair, dominant, straight hair, recessive. Having dimples on the cheek is a dominant character, normal cheeks, recessive. Being right-handed, dominant, left-handed, recessive. Then roller and non-rolling of the tongue, I've told you. There some people can roll their tongue, some can't. So rolling of the tongue is a dominant character, and non-rolling is a recessive character. Then blood group Rh positive. So for being positive is a dominant character, and Rh negative is a recessive character. Then taster then is a dominant character, while the non-tasting is a recessive character. And the normal pigmentation of the skin, if you're having skin pigmentation, that is a dominant character. And the albino people suffering from albinism, you must have seen they don't have skin pigmentation on them. That is a recessive character. So when we study Mendel's monohybrid cross, then you will properly understand what is a dominant character and what is recessive character. Okay, so we'll start with the Mendel's monohybrid cross students. So also you can write it down. You can write the table in order to understand and do the cross. What is monohybrid cross? The cross with only one pair of contrasting characters is called monohybrid cross, right? For example, if you are studying height of the plant, so if you are studying inheritance of the height of the plant, that is tall and dwarf, so this one contrasting character you're taking, so that is called as a monohybrid cross. So you can take another example also. For example, round and wrinkled seed. So if you are studying this character, so round shape of the seed, round, and inheritance of the round and wrinkled seed. If you are studying that in the next generation, what kind of seed is going to be there, and in the next following generation, what kind of seeds are going to be there. So that is called as the monohybrid cross, right? So the cross with only one pair of contrasting characters is called as monohybrid cross. So in this experiment, which we are going to study, so let us consider the characteristic plant height with a pair of contrasting characters, that is tall plant and dwarf plant. So first of all, with this figure, I'll try to explain you what he has said. So what he has done, the Mendel has taken a parental generation. He has taken a tall plant and a dwarf plant, which is small in size, and he has allowed for the self crossing between them. So when he has crossed, then in the next generation, he has found that all the plants, you can see both the plants tall here, right? So in the F1 generation, both the plants were looking tall. So, and then in the when the selfing is done between this type of tall plants, then in the F2 generation, you can see three tall plants and one dwarf plant. That means he has said that the 3:1 ratio is there. So this figure I'm showing you only to show you the height, that is tall and dwarf. Now let us try to understand this with the help of a table. Okay, so the parental generation. Okay, so the parental generation phenotype. What is the phenotype? Okay, I'll use a pointer so that I can point it out. Phenotype. See, in the parental generation, he has taken tall and dark plant. So that is a phenotype. So what is the definition of phenotype? Phenotype means external appearance or visible characteristic of the organism, right? Whatever the external appearance is there, which you can see with your eye, that is called as the phenotype. So what was the phenotype of the parental generation? Uh, One was the tall plant and the other was the dwarf plant. So the genotype, what he has said is, so Mendel has considered that tallness is a dominant character. So in dominant character, you always have to write with the capital letters. You can see here genotype for the dominant characteristic. Tall is a dominant character, so we have used capital letters, that is capital T, capital T, and the genotype for the dwarf is small t, small t. Okay, so gametes, so we know in mitogenesis, that is during the cell division of meiosis. So it is the cells become haploid. So from capital T, two T's, only one gamete will remain, that is T. And out of this dwarf, small t, we will get the gamete small t. Right? When these gametes combine with the capital T and small t, what will be the combination? Capital T and small t. So the first female generation will be capital T, small t, and if even if you're having one capital letter in your table, right? If one capital is there, that means it is the phenotype will be dominant, right? So here phenotype is tall, you can see, right? So what's the genotype? Genotype is the pairs of genes or the factors responsible for the visible character. So these are the genes. Okay, so I hope you have understood this much. So the phenotype of the parental generation, tall and dwarf, were crossed, and in the first filial generation, we have got all the plants tall, right? Now the F2 generation, the selfing is done between F1. That means the plants which Mendel has got in the F1 generation, that means all the tall plants. You can see that plants were all tall, but the genotype was different. In the parental generation, the genotype was capital T for dominant. But here, though the phenotype is tall, but here the genotype is capital T and small t. So the selfing was done between these tall plants. So the genotypes are capital T, small t, capital T, small t. So the gametes are capital T and small t, and the gametes are capital T and small t. So this is the second filial generation. You can see you have to write, this is how you have to write the Punnett square. This is called as Punnett square. You have to make the table like this. You will write these gametes in the horizontal and the vertical manner. So this capital T, small t, you will write in this way, and here also capital T and small t. So then you have to combine in this manner, this capital T and this capital T, you'll write here. This capital T and small t, you'll write here. Always you have to write the capital letter first. Then this capital T and this small t will be here. And this small t and small t will be here. Right? So you can see here in the table, capital T, capital T. So what will be the phenotype? Tall. Capital T, small t, the phenotype will be tall. Capital T, small t, again phenotype is tall, and the small t, the phenotype is dwarf because it is a recessive character, right? So you can see, Mendel has concluded that in the F2 generation, the phenotypic ratio is 3 is to 1. That means 3 tall and 1 dwarf plant, right? So here, what the genotypic ratio is, see, this is capital T, capital T. This is same like parental generation. This is pure dominant, right? This is called as pure dominant because it is homozygous, and it is also called as homozygous. And these are heterozygous. These two plants, these two, which you can see, capital T, small t, capital T, small t. So they are tall plants, but still their genotype is heterozygous. They are called as hybrid plants, right? And again, this is a pure recessive plant, right? So what were the results of monohybrid cross? So he has found that he crossed a tall plant with a dwarf plant and observed how the triads are transmitted, the progeny, and calculated the percentage of tallness and dwarfness in subsequent generation. When a pure breeding tall plant was crossed with a pure breeding dwarf plant, all the plants were tall in the first filial generation. As we have seen, when such F1 tall plants were allowed to self-pollinate, both the tall and the dwarf plants were appeared in the second generation. Isn't it? We have seen 3 tall and 1 dwarf plant was seen in the next generation. So this you have to keep in mind. According to the data collected by Mendel, so he has planted 929 plants. So out of this 929 pea plants, 705 were tall and 224 were short. Right? So now we have seen that out of these 705, so are all the 705 plants to pure dominant? No. It includes pure dominant as well as mixed kind. That is, it is also heterozygous or hybrid tall plants are also there, and the remaining other pure short plants, right? Pure recessive. So now what is important is the principles laws of heredity. So this is the law. Thus the phenotypic ratio of these plants are 3 tall is to 1 dwarf. That is, the phenotypic ratio will be 3 is to 1. And this is fixed, right? If whenever you will solve the Mendel's monohybrid cross, the phenotypic ratio, whatever characteristic you take, the phenotypic ratio will be 3 is to 1. You can see this is called the phenotypic ratio. Phenotypic ratio 3 is to 1, and the phenotypic ratio will be 1 is to 2 is to 1. Right? What is genotypic ratio? 1 was the pure dominant, 2 were the heterozygous, that is hybrid plants, and 1 was the pure recessive. So In Mendel's monohybrid cross, you have to remember phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. Thus, it can be inferred that in the F2 generation, phenotypically there are two types of plants, whereas genotypically there are three types, right? So, I hope all of you have followed the Mendel's monohybrid cross. If you have followed this monohybrid cross, then easily you'll understand the Mendel's dihybrid cross also. Okay, so let us start with the Mendel's dihybrid cross. So again, it is important with respect to your exams and also with respect to your future. If you are taking science field, then in 11th and 12th standard also you'll find this Mendel's monohybrid and dihybrid cross and the details of it. So your basics should be very clear. So Mendel's experiment on dihybrid cross. Mendel brought about a cross between a pea plant producing rounded and yellow colored seeds and a pea plant with wrinkled and green colored seeds. In this cross, two pairs of contrasting characteristics were considered. Which were the two characters now? Color of the seed and the shape of the seed. Hence, it is called as dihybrid cross. Right? Now, we are going to study two characters at a time. See, the one is round yellow seed. Round is the shape, yellow is the color, and next is the wrinkled green seed. Wrinkled is shape and green is the color. So shape and color, two characters at a time he has studied, and that is why it is called as dihybrid cross. Okay, see now the phenotype. Now all of you know very clearly what is phenotype, what is genotype, what is dominant, what is recessive. Right? Phenotype is the external appearance. Genotype are the gametes responsible for the external appearance. And dominant is the character because the character which is expressed in the next generation completely it hides or masks the other character, and that character is not shown at all in the next generation. That is why that character is called as the dominant character, right? So the character which is seen in the first female generation is a dominant character, he has said. So that is dominant character, and the recessive character which is not expressed at all in the first generation, right? So we'll start. So phenotype here is, you can see, round yellow seed he has taken. He has taken the plants with round yellow seed. Also he has taken the plant wrinkled green, and then he has allowed to the self-pollination between them, and then after fertilization. So the genotypes for round yellow seed will be, you can do on your own. What will be the genotype? As round and yellow is a dominant characteristic, so the genotype is capital R, and capital Y, capital Y. And wrinkled green is a recessive character, that is why small r, and small y, small y. Here you don't write small w, and small g, but you have to write the letters opposite to the dominant. So dominant here we have used round and yellow, so R and Y. So here for this you have to use small r and small y. So gametes here you can see R, capital R, capital R and capital Y. And here the gametes will be how this will be small r and small y. Now in the first filial generation, when you'll combine, so you'll see that capital R, small r, capital Y, small y. So these two characters. So what will be the phenotype? You can see as the capital letters are there, that means the dominant character is there. Capital R is round and capital Y is yellow. So he has found, as it was expected. What do you expect? From the Mendel's monohybrid cross, we have learned that in the first filial generation, all the plants with the dominant characters will appear, right? So here you can see, and here Mendel also expected that the first generation will be all round and yellow. So his expectation was correct. So the phenotype was round yellow seeds. So in the first generation, all the plants were observed with the rounded yellow seeds. Right now, though they were having rounded yellow seeds in them, but they have the character of recessiveness also in them. See, you can see small r and small y is also there. It's not pure round yellow seeds. It is a hybrid round yellow seeds, right? So though the phenotype, all the plants were seen with the round yellow seeds, but they had the characters for wrinkled green seeds also in them, which was masked. Then in next generation we'll see. So these round yellow type of seeds, round yellow type of plants were allowed to self-pollinate. He has again done the crossing between. So round yellow seeds were allowed to cross-pollinate with the round yellow seeds. So the genotype you can write now. You can write the genotype what it is. Capital R, small r, and capital Y, small y. And here also the genotype is same. Capital R, capital Y, small y. Now gametes. Now you should be able to write down gametes. Right? How will you write the gametes? Out of this, four types of gametes are possible. That is capital R, capital Y, capital R, small y. Then you'll take small r, capital Y, and small r, small y. In the same way, here also capital R, capital Y, capital R, small y. So first take capital R and capital and small y's, and then you'll take small r and then capital and small y. So these four of gametes are possible. This is important. You should be able to write down the gametes. Okay, again I'll give you capital R, capital Y, then capital R, small y, then small r, capital Y, and then small r, small y. So that's why I've told you to write it down. If you're writing, then you will understand it better. So it will be capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Right? So if you do the practice of this, if you solve this Dihybrid cross in your notebooks at home, solve it 2-3 times and then take the other characteristic and then solve it on your own, then easily you'll be able to do it. Now let us see the second filial generation. So again, this is a Punnett square, and how you have to arrange the gametes. You know that you have to arrange the gametes in a vertical manner and a horizontal manner. These four gametes you'll write in the horizontal manner and also write in vertical manner. Right, now you'll see the combinations. Now this and this combined together will form this. Right, so how you'll write? Capital R, capital R, capital I, capital Y. In this, capital R, capital Y, small y. Then capital R here, capital R and small r, and capital Y, capital Y. Then capital R, small r, capital Y, small y. Right, in the same way you have to go on doing. So the next column, this is capital R, small y, right? And here it is capital R, capital Y. So what the gametes will be capital R, capital R, capital Y, small y. Okay, here next, capital R, capital R. Okay, so you can do that on your own also. Try to do it. So it will be capital R, small y, small y. So when you go on doing this, and you will get the 16 different possibilities. Right, now you have to tell me one by one what type of seeds you will find. So first will be round yellow, isn't it? Because the dominant characters are there. This is also round yellow because capital Y is there. This is also round yellow, round yellow, round yellow. This is round green because recessive Y is there. Can you see? Round green. This will be round yellow. It will be round green again. This is round yellow, round yellow. This is C small r, so it is not round, that is wrinkled, and capital Y, so wrinkled yellow. This is again wrinkled yellow. This is round yellow. Now R is capital, so it is round, and Y is small, so it is green. Right? This is round green. Here small r, so that means it is not round, it is wrinkled, and Y capital, so it is yellow, wrinkled yellow. And then the last one, small r means wrinkled and small y means green, right? So it is wrinkled green. So with the help of the table, before I tell you, you have to calculate the phenotypic ratio and the genotypic ratio, right? So you tell me how many round yellows are there. So we can count how many round yellows. 1, 2, 3, 4, 5, 6, 7, 8, and 9, right? So 9 round yellow seeds are there. Then, which is the dominant character. But also we have found we have got a new hybrid kind of seeds, wrinkled yellow. Now wrinkled yellow seeds were not there in the parent generation, isn't it? We were not having wrinkled yellow. We are having round yellow and wrinkled green. But now we are getting a new combination, that is wrinkled yellow. Where are the wrinkled yellows? Here you can see how many are there. 1, 2, 3. 3 wrinkled yellow. Now round green. Now round green was not there, wrinkled green was there in the parental generation, but now this is a new combination, that is round green. How many round green? 1, 2, 3. Right? Then wrinkled green. This is a parental characteristic. This is wrinkled green. So we have got wrinkled green only one. Right? So can you see the recessive characters are expressed very less. That is why they are called recessive. Right? Here round yellow is the character which is more, so that is why it is called dominant characters. So in this way, Mendel has said dominant and recessive characters. The characters which are expressed more, that those are called as dominant, while the characters which are expressed less are called as recessive characters. So here the phenotypic ratio is of round yellow, wrinkled yellow, round green, and wrinkled green. So the phenotypic ratio is round yellow 9, wrinkled yellow 3, round green 3, and wrinkled green 1. So the phenotypic ratio for the Mendel's dihybrid crosses 9 is to 3 is to 3 is to 1. And the genotype ratio, first you have to write what are the different types of genotypes which you have got. So we have got first capital R, capital R, capital Y, capital Y. We have got this one genotype. One plant was with this genotype. This is a pure dominant plant, as like the parental generation P1. This is pure dominant. Now it is left only one. The rest all have become hybrid. Then capital R, capital R, capital Y, capital Y. Now here this is also round yellow, but again this is hybrid. So it is heterozygous. So two plants were like this. Then the third genotype which you can see in the table is capital R, small y, it is 1. Then capital R, small r, capital Y, there are 2. Capital R, capital Y, small y are 4. Capital R, small y are 2. From the table, all this you have to write down. So small r, capital Y is 1. And small r, capital Y, small y is 2, and small y is 1, that is pure recessive. So how many type of how many different types of genotypes you can see? 9. Can you see? 9 different types of combinations we have got in the next generations. So though the phenotypic ratio was only 4, here the genotypic ratios are 9. So that is how the variations are seen. So this is not only in plants but also in animals and human beings. That is how we are getting the variations. So we say that what do you say? The offsprings are similar to their parents, but they have variations also. So this is how the variations come, right? So though we may have the same kind of phenotype similar to our parents, but still we might have inside us the different kind of genotypes, which is expressed in a different way in the next generation, right? So this is responsible. This is called as variation, which Mendel has studied. Right, so here the genotypic ratio is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. Right, so here the Mendel has given us the answer that how, though we are similar, still we are different. Right, though there are so many millions of human beings are there on the earth, but still there are variations in their faces, in their appearance. It is because of this. It is because of such kind of different types of combinations. In every next generation, the genotypes changes. So again, now in exams, this is an important question. Distinguish between monohybrid and dihybrid cross. Now, after so much explanation, I think it should be easy for you to write the differentiation between. So you can see monohybrid cross, inheritance of a single allele pair. Allele pair means inheritance of a single contrasting character. And in dihybrid cross, inheritance of the two pairs of contrasting characters. So Mendel's monohybrid class defined in Mendel's first law, and here it is defined in Mendel's second law. Then the phenotypic ratio, this you have to remember, these are important. The phenotypic ratio in the F2 generation in monohybrid cross is 3 is to 1, while the phenotypic ratio of the F2 generation in the hybrid cross is 9 is to 3 is to 1. Genotypic ratio in F2 generation is 1 is to 2 is to 1, while here the genotypic ratio is 1 is 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. This to remember, there is no other option. You have to learn this. Then used to this is monohybrid cross is used to study the pattern of inheritance during first and second generation. And here it is used to study the inheritance pattern of dominant and recessive characters for two different triads. Right? So you can write the distinguish between in a simple language also on your own. The 4 points are expected for 2 marks. Okay, so before we move on to the assignment work, we will revise quickly. So what we have studied today, we have studied about Mendel, and we have studied in details the Mendel's monohybrid cross and Mendel's dihybrid cross. I hope you have followed, right? So now here is an assignment for you. You can take a screenshot and or write it down. Explain Mendel's monohybrid progeny with the help of a contrasting character round and wrinkled seed by Punnett square method. So now we have studied the monohybrid cross with tall and dwarf plant, but here you have to solve this monohybrid cross for the round and wrinkled seed character. Okay, and also you have to explain Mendel's dihybrid cross with the help of any one cross, right? So uh, we know the table, it is there in the textbook also. on page number 184 all the different contrasting 7 mutually contrasting characteristics. So you can take any two characteristics of the plant and solve the Mendel's dihybrid cross in your notebooks. Okay, so we'll stop here. I hope you have followed. So if you're having any doubts, you can ask me.